So we're continuing on with pathology and entomology here. So once again, the autopsy, it basically means to see with one's own eyes, and not all deaths result in autopsy. It all depends upon the circumstances. You've got two basic types of autopsies, forensic and medical. Your forensic autopsy is performed by a medical examiner, usually a forensic pathologist, does not require a family's consent, and usually done because there's a sudden unexpected death, a violent death, or an unattended or suspicious death. Say they find a body and the body has been dead in a house for a while and no one knows what's going on there. Medical autopsy is performed by a pathologist, requires the family's consent. It determines the extent of a disease, the effects of a therapy possibly. So maybe if the person is doing some type of research, drug research because of a cancer, and they're trying to determine whether or not what that drug has done to the body, they would do a medical autopsy to see the effects of the drug on the possible disease. They would look at the presence of any undiagnosed disease. And basically, and fewer than 12% of any regular deaths are actually autopsied. The role of forensic pathologists is to look at the cause of death, the mechanisms of death, the manner of death, the time of death, and sometimes to figure out the identity of the deceased. There's some famous ones out there. Michael Baden, we've seen some shows with that. Dr. G, she's on Discovery Channel. We watched one video. One you probably don't recognize is the Quincy one. That's the one I actually grew up with in the 70s. That's Jack Klugman there. Patricia Cornwell, she actually never was a pathologist. She actually worked and helped pathologists in the office, and then she ended up writing crime books, and her crime books are really well known. And the one I actually don't know anything about, which is Crossing Jordan, but apparently it's pretty well known. So to become a forensic pathologist, you have to have 4 years of college where you earn your BS, 4 years of medical school when you earn an MD, 4 to 5 years of residency, so you become eligible to take your board of exams in pathology, and 1 to 2 years of specialty forensic pathology fellowship. So if you think about it, once you're 18, you have yourself here, if you're lucky and nothing goes wrong, 12, 13 years of school past your time when you're 18 to do this. And it could go on to like 14 years. So it's a good long time, and you're not making a whole lot of money during this time. And you are adding up the money for your medical school and your training. So one of the things that you are doing is determining time of death. Done most accurately if the body is found within the first 24 hours of death. And some of the early indicators we've talked about in class already are livor mortis, algor mortis, rigor mortis, looking at the stomach contents and potassium levels in the eye fluids. Later indicators about 1 to 2 days have passed, decomposition, and insect action, which is the entomology I was talking about. Rigor mortis is the rigidity of the skeletal muscles after death. The onset is as soon as 10 minutes if there's been a lot of exercising going on, to about 2 hours. Your small muscles, your facial muscles are affected first and then spreads throughout the rest of your body. Your maximum stiffness reaches between 12 and 24 hours past the 12 to 24 hours post-mortem, and the joints are stiff for about 1 to 3 days, and then ions leak out, causing your muscles finally to relax. Here's some kind of biology and chemistry of it if you have an interest in it. Those of you that are working on your AP stuff on this, it may be of an interest for you. Livor mortis is settling of the blood resulting in this red-purple color pattern, the color of death, that it's known. You can see a picture of there at the back of it. It looks like major bruising going on. As the body decomposes, blood seeps through the tissue and settles at the lowest point. So it's actually one of the ways you can kind of figure out where the body has been settled at. The body is that blood is going to kind of fall to that portion. It begins about 2 hours after death and becomes set between 8 to 12 hours after death. It can indicate the position of the body after death or if they were moved. Algor mortis is the cooling rate of the body after death, the chill of death. And this is something we've actually did some work on already, some math on it. You can use a rectal temperature or the liver temperature. Generally, the body cools about 1.4 degrees Fahrenheit an hour for the first 12 hours and then 0.7 degrees Fahrenheit after that point. And it can be influenced anything from the weather to amount of fat on the individual to drugs in the body to clothing. So this right here is an example problem, which we've already done a bunch of those problems in class. More fun-filled example problems, which, like I said, we've done in class. Some other things that your pathologist is going to look at is your stomach contents, because we know that food actually breaks down at a fairly predictable rate. It's not exactly perfectly accurate because you can have a lot of other things going on. You can have drugs in your system. Everybody's metabolism is a little bit different. I know when I was younger, I had an exceptionally fast metabolism. Some people don't. Some food goes slower than others. Were you exercising? All those things can actually make things go slower or faster. And the other thing is they look in your eyes. The vitreous humor, the potassium levels increase in the fluid of your eye after death. It's relatively stable, less susceptible than other body fluids to rapid chemical changes and contamination. We've done some work on the body farm. In fact, you basically read a book about the guy Dr. Bass, who kind of started the whole body farm idea. It's a nickname of 2.5 acre research facility at the University of Tennessee, and it was developed in 1980 by Bill Bass. And bodies are placed in various conditions and are allowed to decompose, and the idea is to actually see what goes on. In notes earlier, we did talk about the 5 main stages of decay. Some give it a little bit more, some give it a little bit less. But some of the things that can affect those, the affected decay are temperature. Here in the south, things go a lot faster with the humidity and the temperature. Can the insects get to the body? And how far, if the body's been buried or tried to, hiding it, how deeply was it buried? They can also take a look at the insects and look at the stages those insects are in to give an idea of what's going on. I'm not even talking about the life cycle of a particular insect that is looked at, but also which insects are there, because insects come at different points in the decay stage of the body. Four common flies that are used in forensic investigation. You got your house fly, your blowfly, which is real famous. There's a lot of books that actually have the title blowfly in it. Your skipper fly, and something disgustingly called the flesh fly. And here's your blowfly life cycle. It's very much temperature dependent. So you've got the time here for you. So you've got basically your little fly there. She's going to lay her eggs. After 22 hours, you're going to get basically a larva in 27 hours. And then 22 hours after that. Then 130 hours. And then, woof, you got your fly again. All right, when you're looking at decay stages, here's your first stage, fresh or initial decay, 0 to 3 days after death. On the body, the bacteria in the body's own digestive enzymes begin the whole decay situation. On the outside, flies are attracted to the corpse and lay eggs around wounds and natural body openings. So you're looking at your nose, your ears, your mouth, your anus, any of those fun-filled, exciting areas that are open. And the eggs hatch and move into the bodies. So you get the blowfly right there. Stage 2, you got your bloat and putrefaction. That's 4 to 10 days after death. And the inside, you got swelling occurring due to the gases produced by the bacteria. That's putrefaction. On the outside, flies and maggots are feeding in great quantity. They're having a buffet. Stage 3, you've got decay and black putrefaction. That's 10 to 20 days after death. On the inside of the body, the skin breaks and gases escape. Fluid leaks out, getting kind of messy now. Exposed parts are black in color. On the outside, maggots are large. They've had a great time in dinner, and there are lots of them, and you've got some massive odor going on. Stage 4 is your post-decay. It's 20 to 50 days after death. On the inside, you got mostly hair, skin, and bones. On the outside, most flies are gone because you don't have the soft, delicious flesh for them anymore. And at this point, you got beetles coming in. They're feeding on the skin and the ligaments. They're having their buffet. Stage 5 is dry and skeletal. This is 50 to 365 days after death. And you notice there's a huge kind of distance, 50 and 365. I mean, this also has a lot to do with temperature, once again, moisture levels, how and what they're buried. So you've got a pretty good amount of possibilities here. On the inside, the body is dry, all the hair disappears, and you got bones only. On the outside, the moths are feeding on the hair. So there is still a buffet going on. Now, this is going to be a whole other section for us. It's forensic anthropology, studying on the bones here. So I am not going to, I might go through this a little bit. Let me see here. No, we will actually hit this section probably in our next area. We'll talk about forensic anthropology, because forensic anthropology is its own unit. There's so much going on with that. You think pathologists and anthropologists do the same thing, but pathologists really look at the flesh. Anthropologists look at the bones. It's a whole different field, and they do a little bit of a little bit different, and sometimes even a little bit more a variety of things.